Here. All right. So this series of lectures are going to be looking at evolution and diversity. So what I want to emphasize is to you is the importance of microbial evolution and how microbial evolution has been the driving force that has contributed to the great diversity that we have in microorganisms. And again, when we think about systemics and evolution, I want you to remember that evolution is the foundation of biology because it is going to be the study of the biodiversity that we have. And we're concentrating mostly on the biodiversity of microorganisms, which are among the most diverse organisms that are present in our planet. And it also helps us with the idea that once we understand that diversity, we can classify it. And classifying the organisms, it's going to be beneficial for us to study them, but also, since you know a lot of microorganisms are pathogenic, to be able to understand which are the ones which are causing havoc, and therefore be able to respond to threats that are generated by microorganisms. And it's going to also give us insight into the process itself, the process of evolution. How does a species become a species? How does that species become a species after they adapt to their new environments? And those adaptations could be by natural selection or artificial selection. And we're going to talk about that as well. And again, it's actually really fun to learn about how organisms come up in the universe. So intellectually and aesthetically, it's actually a very emotional, it's very exciting science. So what I'm going to start to do is to begin by giving you an overview, most likely you have seen this before, about the origins of life. And again, this is just to set up the framework to remember that microorganisms have been here longer than we have, and they are the ones responsible for the world as we know it, in great part. And as you know, when we think about this from figure 12.1, we can see that the formation of the planet Earth and the environment that was present when the planet was formed was very hostile to the life as we know it today. The environment was very anoxic. There was no oxygen present in the planet as we know it. The temperatures were very high, most likely a lot of pressure, a lot of different gases that made life as we know it today inhospitable. So hotter and anoxic was the issue. So it is believed that the first attempts to life happened biochemically in a very abiotic environment. And that is what is shown over here in this graph. So what we're looking at is that before the origin of life, we have a prebiotic chemical lab where you have, let's example, molecules that were interacting with one another. So as I was saying, it's believed that the first form of life was chemical in nature. And eventually, we started to see the emergence of what is believed to have been catalytic RNAs. So the world was based on RNA, and those RNA molecules were self-replicating. And from there, everything began to take off. Those RNAs serve as the catalysis, so they were enzymatic in nature, but they also were the repository of the information. Eventually, that led to the formation of DNA as the molecule that held the information, and from that point, the early cell came up. Now, you have an early cell that is growing in an anoxic environment, most likely now with hydrogen present in the atmosphere, very high temperature. So the most primitive microorganism that we understand that may have come was one that also were living in a very inhospitable environment, not like the one that we know today. And we know this from the fossil record. If we look at some structures called stromatolites, stromatolites are fossilized microbial communities that are the combination of the microbes and sediment that get together. And the stromatolites still exist today. Some of them as the original fossilized stromatolites, like the one here in A and in B. You can see the multiple lines in the stromatolite, and those are considered to be, just like in the tree, those lines are identified as timelines. But when you look at those stromatolites, they were full of filamentous microorganisms that most likely were growing in the absence of oxygen based on the information that we had. And we can compare those ancient stromatolites to the stromatolites that we have now, because they are still existent and there are now some of them that are completely alive and well. And the ones that we have today, we believe, you know what? They have prototrophic bacteria, bacteria that are using light to harness energy. And now they have cyanobacteria or green non-sulfur bacteria. And those we can still see in the ancient stromatolites. So depending on where you look in the stromatolites on the fossil record, you're going to eventually see that some of them were very ancient. We still have a lot of problems finding fossilized microorganisms because they don't fossilize as well as, for example, the dinosaurs that we all love to see the bones because they're too tiny. And then you have to also do microscopic analysis. It's not like you can find a pair of bones and say, you know what, there is a fossil there. You have to do more involved research. So it is believed then that the microorganisms for communities, those communities dependent on one another, and eventually those communities evolve as the selective pressures that were present at the environment and at the time shaped their evolution. Now, we believe that since there was no oxygen, that they may have had a very primitive system of energy derivation. And that energy derivation may have come up from, for example, using hydrogen and sulfur as energy and electron sources. So this picture that is here in the graph, it's showing an ancient, what we believe could be an ancient anaerobic chemolithotrophic bacteria, in which hydrogen that is generated from the reaction of having iron sulfide with hydrogen sulfide generated hydrogen disulfide, iron disulfide, and from there you also now have hydrogen. The hydrogen could have been pumped across a primitive hydrogenase that will then donate those electrons in. The electrons from hydrogen, will be donated by the primitive hydrogenase, and those electrons could then be able to reduce sulfur into more hydrogen sulfide, which will later help catalyze the reaction again. And eventually, that proton gradient that could have been generated by the when hydrogen donated the electrons could have fueled a primitive ATPase, which could have helped with the generation of ATP. Again, this is all hypothesis. We don't have an evidence that this is happening, but we do have organisms which are primitive now in our microbial record that display this kind of respiratory chain. So eventually, with the evolution of cyanobacteria, the bacteria that were able to now have oxygenase photosynthesis, meaning that oxygen is going to be the product of the photosynthetic process, and those bacteria will generate oxygen, and slowly, as you can see here, it begins what is considered to be the great oxidation event. The microorganisms are starting to photosynthesize, splitting water molecules, getting the electrons from water, getting then oxygen as a result of that reaction, and slowly but steadily that began to oxygenize the atmosphere. And when you oxygenize the atmosphere, we can see now the fossil record looking at the areas shown over here in this image. Those lines are the corrosion of iron. So you have now oxidized iron that can deposit in certain areas and show you in the fossil record as these lines. So that then changed and put a different kind of environment into the planet. And as you can see, it took millions of years to get the oxygen levels as we know it now. Plants help eventually, but the initial organisms that pioneered the oxygenation of the atmosphere were cyanobacteria. So we believe that at the formation of the Earth didn't have an atmosphere that was conducive to the life as we know it. It was an oxygenic atmosphere that was very hot. The initial microorganisms must have been chemolithotrophic in nature, probably autotrophic because CO2 was present in the atmosphere. And eventually, one of them managed to, because water was an abundant element, compound in the early Earth, one of them managed to devise and evolve a system where it could split water to get electrons And by the splitting of water through photosynthesis to gain electrons, we now have the production of oxygen. Eventually, we have the initial evidence of microcellular eukaryotes coming up. And those, as you know, grew, populated, and later we start to see the arrival of polycellular organisms, multicellular organisms in the planet. So then that brings us to the idea, like, where did these eukaryotes come from? And as you know, we have the theory of endosymbiosis, which indicates that maybe a primitive eukaryote was able to engulf a bacteria, and that bacteria eventually helped that organism thrive by giving it some level of advantage. So here on the left, we have the idea that early, when you look at a phylogenetic tree, you have a node right there, and that node means that the lineage divergent, and we're going to talk about that in more detail. So here in this side, we have the bacteria. To the side, we have the common ancestors of the Eukarya and the Archaea lineage. And eventually, there was a split where Archaea became its own independent domain, and Eukarya became its own independent domain. Now, we may believe that at a certain point there was some level of evolution of the nucleus as the genetic material became greater. There's a lot of speculation about where the nucleus came from. But eventually what we have always learned is that maybe some kind of bacteria was phagocytosed by one of these early eukaryotes, and that gave rise to the mitochondria. And eventually one of those early eukaryotes that now has a mitochondria took one cyanobacteria as a symbiont and now became a primitive alga. But that doesn't answer a lot of the questions, because as you learn, Archaea have different lipids than bacteria and Eukarya. So if the lipid record were to indicate an idea of the evolution, over here in this point, something weird must have happened, that Archaea was able to, if they have a common ancestor, one kind of lipid must have been in the membrane of the other. So where did that lipid evolution may have happened? It's something that is explained now in this other figure over here that we have in the right. And it's the idea that, as you know, bacteria having phospholipids as we know them, Archaea doesn't. When there was that endosymbiotic relationship, the outer lipids would have been Archaea in type. But we know that, from you have learned in Bio 110 and Bio 2, that the mitochondria, as we know it, has a genome, and that there has been exchange of genetic material from the mitochondria to the nucleus, and from the nucleus to the mitochondria. So it is possibly another alternative idea is that some of the genes that are utilized for membrane lipid production from the symbiont were now adapted by the host, and therefore now they lost the capacity to make the membranes of Archaea, but gained the capacity to make the membrane of bacteria. And that early progenitor now has bacteria-like membrane and not Archaea-like membranes. And that is something that jives with the fossil record that all eukaryotes and all bacteria has the same membrane type. So this is what is shown in this image over here. So that the Archaea continue having their membrane lipids as they have always have them, use of isoprenyl polymers, and bacteria with actually fatty acids and all the construction of phospholipids. So what we also have over here is the idea that maybe you had a bacteria that produced hydrogen, and that the Archaea needed hydrogen for its metabolic activity, and that was the initial part of the relationship, that one was able to utilize hydrogen, produce hydrogen biochemically, and the other one utilized it. Okay? Any questions so far? All right. So this is the initial idea about how the organelles, the mitochondria and the chloroplasts, are believed to have come to life. So now let's try to think about how do we establish the relationship between these microorganisms and basically every organism to one another. And that is the study of phylogeny. So that's what we're going to be looking at right now. So phylogeny, when we think about phylogeny, those of you who have taken microbial evolution or evolution period, you're trying to understand the evolutionary history among organisms. And there was a lot of attempts to do phylogeny. And phylogeny could be established by morphology, for example. When do quadruped animals came up? So we can organize all the animals from the initial phylogenetic announced. You have this group of animals called the Monera, which basically were everything that was a microbe, including protists. And from there, you have the Protista later come up, and from there, plants, animals, and fungi. So that was very basic level of phylogeny. But that didn't explain the relationship of these organisms. It just was used with the tools that the scientists had at the time, which was basically morphological. Now, Carl Woos at the University of Illinois, my undergraduate school, when I was there, was already beginning to try to figure out how do we relate all the organisms to one another. And what he basically did at that point was the idea that you can look at what is called a molecular chronometer. And a molecular chronometer, it's basically a molecule that you can understand and study it across evolutionary time. So that chronometer has to have one particular set of qualities to be able to be a good evolutionary molecular chronometer. It has to be universally distributed, meaning that every organism must have it. It must be functionally constant, meaning that every organism must have the same function for that same molecule. Highly conserved, meaning that it's going to be, even when you have evolutionary changes by mutation happening, that you're not going to diverge into a new molecule. So that maintains a pressure to be conserved as a functional molecule. And that the gene has to be long enough to be able to compare it among different organisms. And that is when they started to look at the 16S small ribosomal RNA subunit. Why? Because every organism makes proteins. And therefore, all of them are going to have a ribosome, and all of them are going to have a ribosome with a small subunit. And that small subunit has a 16S RNA molecule in it. So all of them have that. It is conserved in the function. All of them are using it for the same purpose. And it's highly conserved. We're going to look at the conservation. Maybe there's parts that are going to be different, as shown over here in this image. When you look at it, is that the structure, the secondary structure of the 16S RNA of bacteria, shown here by E. coli, and I put both of the E. coli molecules here next to one another to show you how the structure is this. Well, this is the same because it's the same organism, but look at the Archaea 16S RNA. The loops, the secondary structure of the RNA, it's all the same compared to the bacterial 16S RNA. Now you know that the ribosomes in eukaryotes are different. They're larger. They don't have an 18S RNA subunit. But when you look at the homologous molecule, that is the 18S RNA, you can appreciate here with the one from Saccharomyces cerevisiae that the structure is similar, that the arrangement is the same. So when you look at it, the 16S RNA was a compelling molecule in which now by the power of sequencing, you can study if it is similar in all organisms and then try to understand how that similarity reflects with one another. So what you can do now is then compare the sequences of that small subunit of RNA and determine the evolutionary relationship between them. Here in color, what is being shown are the regions that are the most variable in bacteria. And as you can see in my other image, this comes from a different book, the images are shown in red. Those red images are showing again the variable region in Archaea as well as bacteria. So a molecular chronometer could be any molecule that could be used to study and then compare how that molecule has evolved in different organisms and then rank them according to the level of similarity between those molecules. So how do we do that? We take the microorganism, we isolate its DNA. That DNA then gets amplified by PCR. And after you amplify it by PCR, you sequence that DNA. And once you now get the sequence, you're able then to align the sequence together. So for example, in this image over here, notice the places where the sequences for 3 different organisms has been aligned. G is common in the first position, a G is common in the second one, and A is common in the third, but now you have to see variation in the 4th, 5th, and 6th position. So if you now put them together and just analyze them straight directly, that may give you a false idea of how similar the sequences are. But then you have to align the sequences correctly next to one another, because what alignment is indicating is that maybe some DNA was inserted into the sequence, or some DNA was removed from the sequence. And therefore, when you do that, you can now appreciate a much better relationship between the sequences. So since the first pair of bases are matched to one another, they align perfectly. But see, maybe this triple A over here was inserted from a different organism. And now this other part was also inserted, and this is actually perfectly aligned with the second sequence below. So you can appreciate that the alignment is extremely important to get a true comparison of the sequences. And now what you see over here in the right are the matrix of the differences between the sequence. Prior to alignment, sequence 1 and sequence 2 had 11 differences between each other. And sequence 1 and 3 has 6 differences between one another. 2 and 3 had 11 differences. But after the appropriate alignment of the sequence, and you do now the matrix, notice that now that difference between sequence 2 and sequence 3 went from 11 to 3, giving you a better idea of the actual relationship between the two. And the alignment eliminated the differences from sequence 3 and sequence 1, from 6 to 0. So definitely having an algorithm that is going to help you align the sequence is extremely important to get the information right. From that analysis, you can form the phylogenetic tree. And the phylogenetic tree is basically a visualization of the relationship between the organisms based on the differences of the sequences. You can have a tree that is unrooted, like this one on top over here that I'm surrounded by my mouse, and that tree is unrooted because you have taken away what we consider a common ancestor. Or you can have the tree rooted, and as the tree rooted, you can see this initial portion over here and there. That is showing you the ancestor to all the organisms that you're sequencing and analyzing together. So either you have the unrooted, and that is just looking at the relationship of the organisms to each other, or you can see it to the relationship they have to an ancestor. Now, the points that branch up are called the nodes. Here are the branches, and at the end, you have the taxons, basically the organisms. So you can compare, like in this image, 7 different taxa and see how they relate to one another. The tree, if we look at taxa from 1, 2, and 3, those 3 organisms, you can see that there's a branching point in here that then branch up again from one common ancestor, organism 1, and an ancestor to organism 2 and 3 arose, and then another ancestor to organism 2 and 3 branch into now organism 2 and 3. So you can see that 2 and 3 are more closely related because they have a common ancestor that is not shared with 1, and definitely not shared with number 7 all the way down here. And then you can see that organisms 1, 2, and 3 are more closely related to one another than to organism number 7, because they share a common ancestor. So what Carl Woese did in his lab was to sequence the 16S RNA of all the organisms that they could. And what they ended up having was the following phylogenetic tree that now look very different from the ones that Whitaker, for example, have put forward. And in this tree, it was evident that you had an initial ancestor, which you call Luca, not because he was in the second floor and you didn't hear him. One person got the joke. Okay, never mind. But because that ancestor eventually branched into what is now considered the domain of bacteria, And later, from there, the other ancestor branched into what is now seen as the domain of Archaea and Eukarya. So this kind of phylogenetic analysis came up with the theory that now we have only 3 domains as opposed to the 5 domains that Whitaker was, for example, showing before. The Monera, Protista, fungi, plant, and animals. When we look at where the position of us, quote unquote, are, we are over here in this tiny branch. Fungi and animals are over here. Everything else is a microorganism now in this particular branch. So the 16S small subunit ribosome was instrumental in now making a better understanding of the phylogeny and showing the relationship that we have among microorganisms. And the beautiful thing about this is that this is supported by biochemical data. When we, for example, look at the homology, we can see, for example, that, except with the issue of the lipid chemistry, that eukaryotes and prokaryotes have different cell walls, for example. That the cell wall of bacteria and archaea is very similar, maybe with a change in the peptidoglycan layer, as you know. Molecularly speaking, bacteria share qualities with themselves that archaea and eukaryotes share together. For example, let me take you over here to this Venn diagram, which is now looking at the genomic analysis and transcription and translation. And I exhort you strongly to actually look at every one of these items in this figure and understand what they mean and what is the relationship to it. So I'm just going to illustrate a couple of them for you so you can get a better idea. So for example, if we look at the structure of the chromosome, the idea that the chromosome is circular versus linear, that trait, it's shared between archaea and bacteria, but not eukarya. If we look about introns, introns are only present in archaea and in eukaryotes and not bacteria. So that indicates that idea of the segregation of the lineages between archaea and eukarya. Centrosomes are only found in eukarya. They're not present in bacteria or archaea. And something like the idea that genome is a double-stranded DNA genome, it's a common trait between every 3 of the domains. So when you look at this figure, you can now take a moment to explore every one of those 20 points and see where they fall and try to now think about them from the point of the phylogeny that gave rise to the 3 domain of life theory. Now, for example, if we look now at the general features of the 3 domains, we can put them in this graph format as shown over here. So, I mean, I don't want to go through every single one of them because, I mean, you can study the table on your own, but I want to point some of them directly to you. So, for example, let's look at the sensitivity to certain antibiotics. That's an important one because most of the antibiotics that we're using now are targeting ribosomes and therefore translation. So when we look at chloramphenicol, streptomycin, kanamycin, and penicillin, penicillin, as you know, is an antibiotic against the cell wall of bacteria, so it's not a ribosome one. Bacteria are sensitive to every single one of those antibiotics, indicating that the machinery that those antibiotics are targeting is exclusive of bacteria. But notice that archaea doesn't have a sensitivity to them, neither does eukarya. Because when we now start to look at the ribosomes, for example, of archaea and eukarya, even though they still are different by size, structure-wise, they're very similar. So the antibiotics that can inhibit translation in bacteria cannot inhibit translation in eukarya or archaea, indicating again, supporting that idea that it's part of that branching that occurred early into an archaea and bacteria different lineage, and that the archaea lineage eventually had a common ancestor with the eukarya. For example, metabolically speaking, if you look at these physiological special structures in the left, Few of any of the eukarya do any of these reactions. They cannot dissimulate sulfur or iron, whereas archaea and bacteria can. They cannot denitrify. Bacteria usually can, archaea cannot. So that is one of the things that probably it was a relationship that was much earlier before the split. So again, take a look at this table, at every single one of those facets, and try to put them together within the contents of the evolution of the microorganisms into the three domains. All right? Okay. Let's now look at the next part of the lecture. If you have any questions. Derek? No? All right. Okay, let's move forward then. So what we're going to look now that we understand the great diversity, phylogenetic diversity of bacteria, archaea, and eukarya, let's take a moment to look at how that diversity was generated and what is driving it. And that is the issue of evolution and natural selection. And that's where I'm going to go next. So one of the things that I want to bring out to you, it's the idea that you have four general mechanisms that are going to generate evolution. And the first one of them you already are very familiar with, and that is mutation. As you know, a mutation is a change in the genome of an organism that is inherited. So that is going to be able to be passed into the offspring of the organism. And those changes can happen really simply by having a substitution, a G being changed for an A, a T, being changed for a C, whatever. A substitution in a nucleotide in the DNA of the organism, or RNA, whatever the genome may be. But you could also have deletions, where pieces of the genome are excised out of the genome and lost. You can have insertion, where now that piece may actually now go somewhere else, and by transposition or recombination be part of a different gene. And last but not least, you have duplications, when one gene can now become two. So those are the forces that are driving mutation. And again, I'm not going to talk yet about what are the causes of mutation and what is driving this, but you remember that the mutations are going to be one of the driving forces that generate evolution. But the thing is that even if they are, most mutations don't have a phenotype. They are considered neutral because of the degenerate nature of the genetic code. Since one amino acid can be encoded oftentimes by 4 different codons, changing one base in the genome may result in another codon that is going to encode the same amino acid. So even though you have a mutation in the genome itself, that doesn't translate to a change on the amino acid. And even if there is a change in the amino acid, you can have a change, for example, of a leucine for an isoleucine. It's still a hydrophobic amino acid. You're not going to have that much of an effect in the overall protein structure or of the structure that you're looking at. We also are going to notice that some mutations are going to be deleterious. You may eliminate a functional gene that is required for survival, and therefore that microorganism dies. But the important part is that a lot of times those mutations are going to be beneficial, and the organisms are going to derive some level, we're going to talk about fitness, by earning those mutations. Now, another part that we're going to talk is genetic flow, and that is going to be the introduction of genes into a population or the loss of those genes from the population. So flowing, how is that gene coming in and out of a population? And genetic flow could happen by what we call now, okay, Marcos, you are blanking. You see when professors have mental fart? By homologous genes transfer, HMC. Now, the next part about this, that is the theory that driven Darwin forward, is the idea of selection. Selection, it's now defined it's defined when it comes to the fitness. You have an ability for an organism to reproduce and pass and contribute to genetic makeup of the generations in the future. So fitness can be evaluated as survival. We always think about the idea of survival of the fittest. So if an organism has gotten a mutation, that mutation has increased its fitness, therefore that organism is now able to reproduce and pass those mutations to its progeny. The other important aspect is genetic drift. And genetic drift, it's a change that is happening in a population, but it's a random change, and that is going to increase the frequency of an allele. We do not think of selection because this is happening as evolution without it. Just because for some reason something happened, that organism now is able to reproduce really well, and now it dominates a particular niche, and therefore the allele that was present in that organism took over. So that is the difference between genetic drift and selection. The absence of, that you get evolution without the selective pressure in genetic drift. So this image over here, it's now showing you an example of how we study, for example, genetic drift in the lab. Here what we have represented in 4 different colors are 4 different microorganisms that, or 4 strains of microorganisms that had an ancestor tube, and as you can appreciate, all of them are represented at the same frequency. Each of them have 25% of the population. We have 20 different creatures in there, 20 different organisms, and each of them is represented by having 5 members in the population, so 25%. Now, by a random process, we can take a sample and put it in 3 different tubes. But since you're taking the sample randomly, the first tube gets 2 yellow, 1 blue, and 1 red. The second tube is going to get 2 red, 1 yellow, and 1 green. And the third tube is going to get 2 reds, 1 yellow, and 1 blue. Now you let the microorganisms divide. And again, you're not changing the environment for them. This is the same environment. They just have a brand new environment. They now are colonizing that environment. And because of the sampling that was taken randomly at the beginning, now when you allow those organisms to populate and now you have them at full growth, you can see that in the first tube here, population number 1, when they grow, now the yellow ones are 50%. They're more than half. When they were a quarter in the original environment, now they are half. Green doesn't exist in this population anymore. So it's 0%. And the red and the blue are now represented as in the same ancestral population at 25%. When you take another sample from that initial population and put it in a new environment, now, instead of catching any drift by random chance, you caught 2 blues and 2 yellows. And therefore, when the tube grew, you have 50% blues and 50% yellows. And eventually, when another event happened, you happen to take all yellows. And therefore, the population now is completely 100% yellow cells. So by genetic drift, with that, in the absence of selection, one population eventually took over that particular environmental niche. And you can see the same process that is happening in the other tubes. At the end, population 2 is all red, and population 3 is 50% yellow, 25% red, 25% blue, but green is now gone from that environment. There was no selection. It's just by the randomness of the system allowing a population to move into a new area and therefore grow. Okay? So if we look now at all these factors, we're looking at mutation, gene flow, selection, and genetic drift. When we look at all of them, we're going to see what is going to be the mechanisms that are going to be mediating natural selection. So I'm going to look into this part. As you know, when we discuss, the genotypes within a population are going to be very variable. And that variability, it's inherited on the offspring of the population. So a mutation in one parent are going to be passed into its offspring. If that parent happens to get a gene from the environment, from horizontal gene transfer, we're going to look at that in a moment, that now it's going to also be passed to their offspring. So you have the variability that is happening due to the mutation and horizontal gene transfer. Do all of you know what horizontal gene transfer is? Okay, Fantastic. Now, you have the organisms. Those organisms have those variabilities that can be passed to their offspring. Now you put in them in an environment, and that environment could have biotic or abiotic forces, we call them selective pressures, that can be acting on them. For example, a biotic environment could have a competitor. So you have one organism competing for the same food as another. You can have a predator. That organism is coming in, and now there's another one that wants to eat it. Or you can have a mutualistic environment where two organisms pair up and their independent strength get combined symbiotically, and they do better than they can do alone. Abiotic influences include the amount of resources present in that environment. It's going to also include the conditions, both physical as well as chemical. So heat, temperature, pH, all the ones that we already have discussed for the organism's environment play a role here, and those are selective pressures. So then you look to see which are the genotypes that are best adapted to and have the better fitness to respond to those selective pressures, and therefore those organisms are the ones which are going to leave offspring. So that is going to be the point of adaptation and fitness. The organism that is better adapted to the environment and has the better fitness and therefore is able to reproduce and leave offspring are the ones who survive the selective pressures. So we have, in the case of microorganisms, we are constantly putting them under selective pressures. What is one of the selective pressures that we put in microorganisms in our daily basis? Antibiotics, well said. So we are constantly putting antibiotics in our system. We now have, that's the selective pressure, the adaptations that those organisms may have, either because they have mutated. Let's think about penicillin. Penicillin targets the FTSI transpeptidase gene. So either the organism can mutate that gene, and therefore that mutation is going to give them a selective advantage, an adaptation to resist penicillin, or is going to now, through horizontal gene transfer, inherit an enzyme that is going to be able to break down the penicillin antibiotic, and therefore also is adapted and it has better fitness. Okay? So microorganisms are not devoid of that selective pressures, and they respond to them in the same way that other organisms by having mutations and horizontal gene transfer that increase their fitness and the way that they can adapt to those pressures. Okay? So now, just to remind you about the idea of horizontal gene transfer, here we have 4 different organisms. Here is an organism and the genome is red, so you can see gene number 2 over here. And by one mechanism or another, we're going to talk about those. It could be transformation, it could be transduction, conjugation, or basically just transfer of a plasmid. That microorganism now is able to get gene 2 into its genome. Here, this other microorganism with the yellow genome is able now to pass gene 3 to the other organism with the green genome. So what you have now is the issue that the genome has a different evolutionary history than that gene. Because the genome as a whole is evolving as its own time clock, but because it has gotten a gene from another organism with a different evolutionary timeline, now that gene has a different history from the entire genome that is surrounding it. And that can mess up really royally when we do phylogeny. Because in phylogeny, we're putting together aligning genes according to their evolutionary history. So in this case, the ancestor to this gene 2 in the bacteria with the blue genome is going to be basically like the blue genome, so it's going to be blue. But when the horizontal gene transfer happens, now it's going to look as the evolution that is going to make it more similar to the organism in red. And that is what is shown in here, in gene number 2, that before the organisms evolve, blue and green were very close to one another, orange and red were very close to one another, but after the horizontal genes transfer, notice that blue and red look like they have evolved together. where it's actually not a result of the evolution of the genome, but the transfer of an evolved gene to that new organism. So that can mess up significantly your phylogeny. The other thing that can mess up your phylogeny are mutations that revert. If you have a gene that has an ATC and it mutates to an ATG and later mutates back to an ATC, you do not have now a record of that mutation because it got erased, because it's now looking like the ancestor. So keep in mind all those points when you're thinking about evolutionary data. All right, let's look at the next point, which the idea of how we study evolution in the lab. And here in this particular part of the issue, I'm looking at a bacteria called Rhodohacer capsulatus, which is a photosynthetic microorganism. It's of the purple bacteria. And in this kind of experiment, what they did is that they took the organism and they put it in conditions of light, therefore where it can have photosynthesis, or in condition of darkness. So let's just put the bacteria to grow. The bacteria can grow in either condition. Now, growing in one condition or another doesn't increase the rate of mutation. The rate of mutation is the same if the bacteria is grown in light or in darkness. The other thing is that the photosynthetic pigments made by the bacteria are made irrespective of the environment. So the bacteria, it's putting all the energy to make those photosynthetic pigments, the bacterial chlorophyll. It's not chlorophyll in bacteria, it's called bacterial chlorophyll. And of course the carotenoids. All the pigments that are designed for photosynthesis are being made by the organisms in the light and in the darkness. Doesn't matter. So now the experiment is looking at subculture numbers. So here we're looking in the left at the amount of bacterial chlorophyll A per ml of culture. So you're measuring how much bacterial chlorophyll is being made. And at the bottom what you have is subculture number. Basically you put a bacteria to grow in your Erlenmeyer flask. It grew. When it gets to stationary point, you transfer more bacteria to the next one and you let it grow. That is a subculture. So you're looking at 5 different passages, 10 different passages, 15 passages, 20 passages. So you're growing the bacteria many generations. It's not generations that you're looking here when you're referring to subculture number. It's how many cultures have been done. And you continue passing them along to allow many generations to happen. So now let's look at the green line, which are the organisms grown in the light. Since you have now light that the organism can use to derive energy, there is a strong selection pressure to keep those genes that are making the light components. A bacteria shown here in white is a mutant. It cannot make bacterial chlorophyll, for example. But notice that even though that bacteria could grow, because it is growing in the absence of light, it doesn't need the light now that it doesn't have those genes, that microorganism is outcompeted by the more fit organisms which can use light to derive energy. So therefore, when you look after many generations, the population of bacteria all have maintained the genes that allow them to do photosynthesis. Now let's take the same culture and put them in the dark. And as you can see, over here, this little microorganism has a mutation for this gene. Both microorganisms are growing in the dark. But it takes energy to make that bacterial chlorophyll and those carotenoids. And therefore, the microorganisms growing in the dark that doesn't spend energy making those pigments is able to grow better. Because it doesn't need light to grow. So it's not wasting resources into making those molecules. And eventually, as you can see, with subsequent generations, that organism that had a mutation for bacterial chlorophyll takes over because it has a better fitness in that environment. There was nothing wrong with the other organism. It was making its genes for bacterial chlorophyll, but it couldn't keep up with the other organism who could grow better because it had probably more energy because it wasn't wasting it making those molecules. So The book also has other examples of evolution in the lab that can be studied. So I strongly suggest that you look at them more carefully so you can see, especially the bacteria, the E. coli genome mutation project, which how now it recently celebrated 50,000 generations of bacteria. So E. coli grown for 50,000 generations, and then looking at how the genome evolved. Any questions about this? All right. So now let's take the last bit of the class to look at systematics. And systematics is considered the study of diversity of microorganisms and their relationship, in particular when we think about taxonomy. So how do we name an organism, and how do we take advantage to distinguish that one organism is indeed a species and that it's not something else? Now, as I mentioned to you before, when we think about species formation, we have to consider the environment of the organism, a selective pressure, and then the random mutation that can happen in an organism, and how the organism uses that mutation to adapt and have better fitness. So here we have a microbiological habitat with 3 different ecotypes. Ecotypes are bacteria that live in an ecosystem. So ecotype 1, 2, and 3, the red, the purple, and the yellow. One microorganism in that yellow population now has a mutation that is adaptive and therefore is going to be beneficial. As the population grows, nothing is happening. The organism is still present, is passing it to its offspring, but now you have a process of selective pressure. That selective pressure is going to allow that organism, now shown in green, to survive. The other one wasn't able to survive. And therefore, that mutant form of ecotype 1 takes over and is able to survive better than the yellow ecotype 1 who was its ancestor. And as you can see, it's now there green as a dominant member and participating in that habitat. What happens later from that is that that process gets repeated over and over again. And eventually, what you have is the generation of a completely new species. Okay? So now, if we have all the species in an environment, we have to then think, how are we cataloging them? And microbiology, as you remember, and we have discussed, has been really good about naming organisms according to phenotypic characteristics. For example, the morphology of the colony, the morphology of the bacteria, Streptococcus, Lactobacillus, Staphylococcus. All those names are based on the morphology. Do they move? Do they have a particular metabolism? What survival do they have in an environment? Halobacterium is named because it's a halophile, which means that it loves high soil concentration. So a lot of the taxonomy terms that we use in microbiology is based on morphology, motility, metabolism, their physiology, lipid chemistry, cell wall chemistry, and another other traits like pigment formation, bioluminescence, antibiotic sensitivity, etc., etc. So we can use all of those to be able to organize microorganisms in groups. And just to remind you about what we do that, we look at the point of taxonomic classification. So we have, thanks to Carl Woese, the 3 domains of life, which is the most encompassing domains that organisms can be classified. Within domains, you have a kingdom. And within that kingdom, you have a phylum. Now, phylums can be classified as classes. And within a class, you have orders. Within orders, you have families. Families, you have genus. And next to genus, you have species. You know the mnemonic to memorize these things? Anybody remember the mnemonic? Go ahead. Oh, that's one. That's new. That's a good one. Dumb King Philip came over for great spaghetti is the one that I know.
I didn't know it was spaghetti.
You know what spaghetti?
It's a different S word.
The one that told me the spaghetti. But yeah, any word that fits your mnemonic will make you remember this. So the whole issue is that you could have, for example, the genus Vibrio. And within Vibrio, you have many species. Vibrio cholera, Vibrio harvey, Vibrio, I'll just name those two for the thing because now the third one, Fisheri, there we go, Vibrio fisheri. So you have that classification. Now, and when you look, for example, at a bacteria, here's Allochromium warmingi, you can look at the domain. The domain is bacteria, the phylum Protobacteria, the class Gamma Protobacteria, the order Stromatolites, the family Chromatacea, the genus Allochromatum, and the species Warmingi. You can also have it, and you can see the properties of each of any one of those. The problem comes that when you're identifying microorganisms, that cannot be the only thing that we can use. Especially now when a lot of microorganisms may be very tightly related to one another. So we can now look at other traits that will allow us to do better identification, and some of those traits are molecular taxonomy. So we have to take a polyphasic approach, basically multiple methods, to be able to take together a microorganism and have a better classification of their relationship with one another. And we use that in particular with bacteria and archaea because you cannot really do the macroscopic approaches that you can do with animals, for example. They're too tiny, we cannot see them very well. So you can then look molecular taxonomy to let you look at the genome. All that information is in their genomes. And therefore you can use it to exploit to check the differences. So for example, you can do multiple gene sequence analysis. So you can take a microorganism, sequence is 16S RNA, you have that, but now take another gene that is important. If you take, for example, the gene for bacterial gyrase, you can then look at how all these microorganisms that are similar by 16S RNA have different phenotype in the gyrase DNA. Or you can use another gene for something super important, like one of the parts of the ribosome, or the protein. So you can look at something that is very conserved within the family. If you're looking at bacteria that are photosynthetic, you can use 16S, and then you can use the gene for bacterial chlorophyll and see how they relate to one another. You can do DNA to DNA hybridization. I'm going to look at that one in particular in a moment. You can do fingerprinting of their genomes, and one technique is ribotyping. I'm not going to have a lot of time to talk about ribotyping, so make sure that you read about it in the textbook. And the other one that I won't have much time to talk about is multi-locus sequence typing too. So you're taking multiple approaches to their genome and comparing them to one another to give you a more detail. You're basically going down, like when you have a microscope and you look at different amplifications, by using these other techniques, you are digging deeper into the branching of the phylogenetic tree that may not be evident just by doing 16S RNA sequence analysis. And the last thing that you can, for example, do is to do a phenotypic analysis. And one of the ones that we do often is to check fatty acids in bacteria because they are very different among themselves. And that is done through a technique called fatty acid methyl ester analysis, or FAME. And it's not the FAME that wants to live forever. One person got the joke. Thank you. Come on, guys, give me a pity laugh. Thank you. All right, there we go. So I'm going to take this one in particular because it's going to help us with the idea of how do we define a species in bacteria and archaea. And what we have here, this is the genomic hybridization as a tool. You take two organisms and you isolate their genomic DNA, and then you shear that genomic DNA into pieces. Now you're going to have a random assortment of pieces of the genome, and with one bacteria you can label that genome with a radioactive probe, for example, P33, which is a radioactive isotope of phosphorus. As you know, the entire backbone of bacteria, of any DNA molecule, it has phosphates. So you use that. So that will be your probe DNA, shown here in green, and now you have your target DNA in red to the side. You put your target DNA on a membrane, you let it stick to the membrane, you block that membrane so you don't have non-specific adhesion of your probe, and then you add your probe to the membrane. And if the sequence of a probe, as you know, it's similar to the sequence of a target, they're going to hybridize. Yes?
What membrane are you talking
It's nitrocellulose. It's a composite membrane that we use in the lab to be able to attach DNA or protein. So natural cellulose. Yeah. So the question was kind of what membrane we're talking about. It's not a cell membrane as we had discussed. It's membrane as in a matrix to bind something to it. So now you hybridize them together. And you look at the amount of hybridization from the probe DNA to the target DNA. And of course, you control it to a hybridization of the probe to itself. That will be 100%. So the result can be seen as percentile. The percentage of hybridization of the probe to the target. And as it says over here, if you have less than 25% hybridization, most likely you're looking at different genera. If you have between 70% and 25, you're looking at bacteria of the same genus, but definitely different species. And if you have greater than 70% hybridization, most likely those bacteria are the same species. But you say 70% is not that much. The same species, shouldn't they have the same hybridization? No. Because as you know, all of us are different. And we are the same species. So even within a species, there is sufficient genetic variation that only you're going to get a respectable 70% hybridization when you compare two different genomes from the same species. Okay? Now, if you combine the 16S RNA sequence similarity with the genome hybridization, you get a heat map plot, not a heat map plot, excuse me, a dot plot like the one shown over here in this image. Here on the left, what we have is the 16S ribosomal RNA gene sequence similarity. So how similar are the two sequences by 16S comparison? And in the bottom part, we have the percent of DNA, genomic DNA hybridization as a percentile as well. So what I want to show you over here are 2 points. As we discussed earlier, here's your 70% gene hybridization. So anything, according to the analysis, that is have a hybridization greater than 70% is the same species. But we can refine that now by looking at the 16S RNA. And when we do that, we are now looking at changes that if the similarity is close to 97% similar and higher, so basically from the pink line up, are supposed to be the same species. So what you can appreciate here, look at this quadrant in the upper right-hand side. That is the quadrant where you have greater than 70% DNA hybridization percentage and greater than 97% 16S hybridization, sequence similarity. That area that is shaded a little darker gray are supposed to be the same species. And as you can appreciate, look at all the different dots. So what do you believe are the different dots about? Why do we have different dots in there if we have the same species? Why are they not more clustered together as a group? You go ahead, Anne.
I would guess that bacteria that are in the same colony are more likely to be closer together because the closeness means that they probably have horizontal gene transfer, so they're more genetically similar.
Right, so microorganisms in a colony are going to be more similar. But remember, this is comparing different organisms. So what are we looking at those dots? What are those dots representing when we're looking? They seem to be by species the same. So they're the same species. Georgina?
Individual differences.
Individual differences. Exactly. And what that we are calling them is the strains. We talk about strains in bacteria. E. coli strain K12. That bacteria is not pathogenic. The strain O150 something, I forgot the exact name, is a pathogenic E. coli. It's the one that is killing people every time there is an outbreak in a hamburger joint. It's the same bacteria, it's E. coli, but one of them is pathogenic and why the other one isn't. That is the idea about what we call the genomes and the pan and the core genome. And that is what is going to be illustrated here in this image. Yes?
So when considering like domain, kingdom, phylum, class, order, so on and so forth, would strains count as like a subspecies?
That's a good question. I will be inclined to think as a strain as a subspecies, but semantically speaking, I'm not sure if an evolutionary biologist will consider a strain and a subspecies the same. I will have to ask my colleagues because I'm not completely sure. I think that a subspecies and a strain could be similar in concept, but I'm not sure if that's the term that an evolutionary biologist will say. Subspecies, strain, the same.
But what would the difference between the two be? Like, what is the difference between subspecies?
Let me then show you, because that is a very good leadway to this idea. So microbiologists decided to take a look at 3 different E. coli strains. And you can do sequence analysis because now sequencing a microorganism is fairly cheap and easy and automated. They just took a sequence analysis of the organisms. And what they found out when they compared the sequence by this kind of map is that, you know what? There's only about 39% of the genes that are shared. Wait a second. They're the same strains. They're the same species. Why is only 39% of the genes that are shared? And right now we have looked at 20 different strains of E. coli. And this tends to kind of hold water for all those 20 strains. That led to the idea, when you put them together in this kind of map, that there is a core set of genes that makes a genome that defines a species. And then whatever is falling out of that core are a set of the pan genome. It's the genomes that encode the core genome plus everything that is not shared within that particular strain. So these are the same species bacteria, different strains. So the pathogenic E. coli versus the E. coli that it's not pathogenic. So why is the pan genome so different from the core genome? Most likely is believed that is the number of genes that has been inherited through horizontal gene transfer. That as microorganisms are picking genes from their environment or being infected by phages that are going to give them new genes, or they mate with one another and get genes that way, that is causing differences in their genome, and that difference is what makes the pan genome what it is, as opposed to all the genes that are the core genome, which define the species. And you had a question.
Yeah, just to clarify, when you do sequence bacterial genome, you're only sequencing their main DNA coil. You're not sequencing the plasmids.
That's exactly right. We're not sequencing. The question was, are we sequencing the genome and the plasmid? No, in this case, it's only the plasmid. But what we're going to learn later in the class is that you can have horizontal gene transfer from the plasmids present, as the plasmid can incorporate themselves into the genome, and when they jump in and out, they can leave pieces of the plasmid in the genome permanently. So that all that kind of issues are going to modify the genome to give a strain, a pan genome that is different from another strain. But together, all those microorganisms are going to share that core genome, and that is what makes them, for example, an E. coli. That core set of genes that it's shared with all of them. Anyway, that's it. I'm just going to do a little acknowledgement here because I got some of my lecture notes from 2 people, Dr. Rasol Chaudhry from Oakland University and Dr. Robert Cox from Dartmouth. So they made their lectures available, so I was able to pick some information and learn from them. So that's it. I'm going to stop the recording over here, and I'll see you on Thursday.